Well, first of all, thank you everyone for coming. And also thank you everyone online for spending time and signing on and listening. I'm Yiping Cao. Just a little bit about me. I came to the States for graduate school. So my undergraduate study is in chemistry. I graduated from UCSB studying mostly applied statistics and molecular microbiology. And then I just became a full-time researcher for over 10 years at Southern California Coastal Water Research Project. And about 2 months ago, Mauricio convinced me to join Source Molecular. And here I am. So today I'd like to share with you, like Mauricio mentioned, a interpretation framework for microbial source tracking data. It's called the Human Fecal Score. But this Human Fecal Score, the framework is general enough to apply to different hosts, different animals. So cow, goat. So collectively, we call them the host fecal score. But I'm going to focus on the human fecal score, because since it's easier to talk about just the human fecal score. But the thing I talk about, it's pretty general, apply to other hosts. So a lot of the MST application work you guys do relates to the purpose of it, relates to some sort of comparison, right? So how does my site compare to other sites? How does this site compare to other sites in my jurisdiction, so that I can prioritize which one I tell the permittees or discharges to focus on, or how does the site in my jurisdiction compare to another jurisdiction, right? Is my site in generally much better? And then another scenario is that how does my site compare to a reference site that has little human activity, right? It's a pretty, compared to that site, it's also pretty good as a reference site. If that's the case, we should all hooray and celebrate, eat cakes. And then, and how does it compare to a site with a known measured health risk through epidemiology study? You can't do epidemiology study at every single site. Those are very pricey, in the millions, like $1 million up to do each epidemiology study. And I already said that compared to another jurisdiction, so you can feel good or bad about your jurisdiction. Then finally, Mauricio already mentioned this. So how does my site look before we implement any BMP remediation actions and after, so that I know whether it's effective or not, was the money spent well? Okay, so how do you answer this? And the best answers usually should be based on solid scientific data through using scientifically sound and statistically defensible approaches. So I put a quotation mark there is because this is actually a quote I'm seeing more and more often in regulatory context. So one example is the 2012 recreational water quality criteria. So the specific language is that we realize we're shifting the regulatory framework from a fixed numeric standard to a risk-based regulatory framework, but you need to demonstrate the control of anthropogenic sources through a scientifically sound and statistically defensible approach. So what does that approach entail? Entails largely 3 parts, right? Do you have the right study design to enable you to collect the right type of data to answer these questions? That's number 1. And then second thing is, do you have a lab who can produce the best quality data, right, on the samples you collect? And then finally, how do you interpret the data to answer the question and in a scientifically, statistically defensible manner? So we'll go forward. Okay, so just elaborate on that a little bit. What really happens when you try to collect micro source tracking data? What generally happens is you send a sampler out, grab some samples from that particular water body you're interested in, you're trying to investigate. You bring the water back, you go through a a suite of steps. You filter the water, then you extract a genetic material from the filter, then you analyze those genetic material for signals from different hosts, right? In the case of human, you have this genetic fragment, a piece of DNA that are from those microbes specifically associated with human, like Mauricio just talked about. Then you generate a big table of data. So what you're seeing here, it's a very simplified version of the data. So you have a bunch of samples. I don't know how many you have. Let's say you have N samples. Each sample will run with some duplication, like in the lab. You always do duplicate to make sure you're getting consistent data. And then this is raw data for qPCR. It's okay, you can just assume these are some numbers. Of course, with any analytical method, you always have non-detect. When you have non-detect, you wouldn't know how much marker you have in your water. So the copy per 100 ml, copy is basically a piece of how many copies of DNA. That's the unit we use for when we say DNA. So we count by 1 cell, 2 cell. For DNA, we say 1 copy, 2 copy. So for these non-detect, of course, you don't know how many copies you have. One very famous saying, it's true, is absence of a detection is not detection of absence. So you can't just say it's 0 because it's non-detect, right? So that's why it's a question mark here. The question mark here, I'll explain a little bit later. They are detected but not quantifiable due to a specific feature to qPCR. I'll explain that later. So really, you have some numbers here. Those are quantifiable results from the raw qPCR data. So now you have these beautiful data from like maybe 30, 100, or 200, 300 samples. How do you make sense of it, right? Because ideally, you want to know what's going on at the site, not with that one individual sample. So the next step is to actually integrate the data, the information, integrate the data from all the samples together, extract information about the site, so you can perform site assessment. And based on that site assessment, you can make management decisions and take action at the site. Okay. So what's this process generally done? So in terms of lab analysis, with all the work that state invested in CBI grant, so the lab process is actually fairly standardized. You know how you do filtration, and state published a manual that's kind of one of the deliverables from the big CBI project, the SIP study. So it basically lays out step by step all the protocols for filtration, DNA extraction, and qPCR. So that middle part is actually fairly standardized. But the front end, that determines whether you're getting the right type of data. You're producing beautiful data here, but is that the right type of data or not? Not so much. And the back end, data integration, not so much either. But how is it done, right? So usually what's done is that you hire a expert or a group of experts for a particular project. So that different project will have different experts, and the expert will help you do the data interpretation and sampling design in some cases. So in the back of your mind, you have these worries, right? So one is the unintentional bias. So is there inherent subjectivity from expert to expert? Am I going to get the same answer if I hire expert A versus expert B? They're all reputable. They publish. They all done this, right? So that's the worry you have. Then another worry is like, what if there's intentional bias, right? What if this person is hired by the discharger? Will they be like more biased towards giving me a conclusion, interpreting the data in a way that's more favorable towards the discharger? And your permittees and dischargers will have the same kind of worry as well. So the expert hired for this project, are they going to be interpreting the data in a way maybe subconsciously more in the favor of regulators? So it can be more stringent in terms of how we're going to be regulated. So these are all legitimate worries. So the question is, are these worries, should we worry? I'll tell you a story before we answer those. Okay. So we all got really curious when we start thinking about this. So what we did is we did a best professional. Oh yeah. So hiring expert interpreting data. That's kind of, we give them a name, a best professional judgment. Okay. So you view the data, you're expert, you judge the data. So we want to find out how consistent or how much variability there really is in best professional judgment. So what we did is, this was published a few years ago, and it's a big group as team efforts, not just me alone. So we got 10 experts together. They are research scientists and water quality managers from across the country. They're from the federal, the research scientists and water quality managers from the federal government, from SCORE, a public research agency, and from academics like universities, and also from a wastewater treatment agency. So we got a 10 experts who have done microresource tracking for a long time. And then what we did is we created a dummy data set for 26 sites. Each site had 20 samples, and then we had simulated data. So everybody got the exact same data set, dummy data, for 26 sites, 26 sets. Then what we asked the expert to do is rank the site from 1 to 6, 1 being the worst contaminated, with regards to how much human contamination they think they have, and then perceived risk, right? Because human, if we assume, so in this case, I'm saying human is the only source. So we asked them to do that. We asked them to do it multiple times. The first time, we said that no talking between you guys. Just do it by yourself. How you gonna do it? Just do it that way. The second time around, when you finish the first time, come back together. Let's talk how you actually did the ranking, right? Let's talk and to see how different we are. Then let's come to a consensus. Let's agree to a common set of principles in terms of how we're gonna rank the site. Then go back and do this ranking again. So how well did the experts do? So what we did is, they're highly variable. So the first time, what we did is that we correlate the ranking among the sites. The correlation coefficient was minus 0.33 to 0.98. It's minus. So that was pretty shocking. What's in here is just a graphic representation. So we're thinking, okay, that's not good. You guys should talk. Let's agree on a common set of principles. So we agreed on a common set of principles. We did again. It definitely improved, as you can see. The blue ones shifted upward, right? The correlation coefficient shifted upward. So the range is, the correlation coefficient is still minus 0.14 to 0.98. The average increased by 0.05, 0.06. So this was shocking to the expert themselves, but also to other people. We actually sent this results to EPA, and there are 2 researchers. One of them is pretty like decades of experience. They were both very shocked. Wow, we had no idea. We know there's variability, but we had no idea. Okay, forward. Oh yeah, so how well did the BPJ work? Not so well in terms of consistency, interpreting, because they came to the conclusion on the exact same data set. That's the kind of really revealing part. And are we right to worry? Yes, probably. We are worried. So next slide. So what came out of this exercise? We published this. So what we came out is like, it's not really that expert not good. It's just that we need it's a subjective way to do this. We need actually a standardized, mathematically defined, objective approach to do this, and that's what's needed as a conclusion from that study. So we assembled a team together, which included myself and Dr. John Griffiths and Dr. Steve Weisberg from SCORE, and also Dr. Oren Shanks, Mano Ganeshan, and Catherine Kelty from US EPA, and Dr. Ali Bing and Dan Wang from Stanford. So we got together and we developed a human fecal score, and it was just published. I think it was online in October last year. It was in print in January. So, so next page. So what exactly is this human fecal score? So it is actually pretty straightforward. It's a site average concentration of the H3 marker. H3 is the best human marker that it has been, ever since it was discovered and published, has gone through so many evaluation studies, cross-continental, national, international, like, so this is like the, yeah, it has always been a winner. But anyway, H113. A lot of you guys already very familiar with this. Forgive me if I'm being redundant here. So it's just average concentration. So what this does is that it's really simple, straightforward, one number. So previously you have all these, right, a big table, and sometimes it's presented as a bunch of graphs, right? You have all these question marks you really don't have a number for, and now you just get a score, 55. So this is pretty straightforward, simple. It's very easy to communicate, and also it's easier for a layman to have a sense of how the scale. It's kind of like when Hilda Bay developed the beach report card, right? And that's one analogy. So when you tell people the concentration, you have 35 enterococcus in the water. Okay, what does that mean? When you tell people, oh, your beach has a grade A, people instantaneously know that's a really great beach. You have F, oh, it's a fail. So people know it's, so that's one of the things. The fecal score is pretty straightforward. It's a simple approach to integrate the data. Not a simple approach, it's a simple representation. It's not simple, it's based on Bayesian models. So okay, go ahead. It's simple, not simplistic, I guess that's the word. So the second thing is that I already said it integrates all data, right? So it's an average. What's the hype about it? Can we just average? But remember, you have a bunch of question marks here. How are you going to average a question mark with the quantifiable data? So previously, the way people deal with it is that, okay, the non-text, maybe we just ignore them. That doesn't seem a very good approach. Okay, if we can't ignore them, let's assign a 0 value to them. That doesn't sound very good either, because remember that absence of detection is not detection absence. So assigning simply a 0 doesn't seem to be right either intuitively. And so that's a non-detect problem. Then there are these detected but not quantifiable. What do we do with it? Like we need a number in order to average. And the quantifiable, that's more straightforward. So previously, you really don't have a way to use all the data together. It's just like a really difficult situation. So the human fecal score, I'll go into details later, but it uses everything. Because even non-detects, there's still information contained in those non-detects, right? If you get like 300 non-detects from a site, that's very good information you have there. You should be able to extract that. Okay, move forward. So in order to understand how the human fecal score actually were able to use all the data, let's take one step back on how qPCR actually quantifies. Okay, I'm going to use an analogy. I use this slide a lot on the current method. So the current method is that you filter the water, you get the little microbes, enterococcus or E. coli on a piece of membrane, then you put the membrane on selective media that these particular, say, let's just use enterococcus like to grow on, selective for enterococcus. Other bacteria are not supposed to grow on it. Then you put it in an incubator at a temperature they like. Happen to 37 of Celsius. So and they'll grow in the incubator overnight. So the cells will start dividing because by itself it's 1 micrometer. You can't see with naked eyes. But once they start dividing, they grow. They form a colony because now they have a population. You can actually see it. The colony actually will change color because there's some specific reagents in this media. They will turn the colony into blue. Now you go and count the colonies. Then you know how many enterococcus you started with in the water. And qPCR are actually very similar, but instead of growing the cells on a petri dish, you're growing a DNA pieces of DNA in a qPCR machine. So say this is a piece of DNA that's specific to the H1N3 bacteria. So, and then we use a polymerase chain reaction, PCR, to basically amplify, just growing it in an exponential rate. So within an hour and a half or 2 hours, 1 copy will turn into millions of copies because exponential. 2 copies turn into 4, 4 turn into 8, and so on and so forth. Each copy of the DNA will emit a little certain amount of fluorescent signal. So as you grow them, so each exponential, each time they grow, it's called a cycle. So one cycle a double, another cycle doubles again and again. So as the population as the copy numbers double, the population doubles. With the cycle you're running, the fluorescence increases. So when it increases until a threshold, it becomes detected by the detector. So the more bacteria you start with, the more DNA you start with, the sooner it's going to get to the threshold and being detected. So that threshold is called cycle of quantification. So as you can tell, this CQ, the cycle of quantification, will be inversely correlated with how much contamination you have in the beginning. Next slide. So this is the basis for qPCR quantification. So you have the CQ, and then you have the log marker concentration. And oh, this thing's supposed to be here. So now you can do that. You can have known concentration of standard material, doing zero dilution, and this is very common in chemistry, and getting a standard curve. Now you have an unknown sample, you go through the same growing the DNA process, you get a CQ value, you just plot it on the standard curve, then you can read how much concentration you had. So it's very similar. So when your environmental when the concentration of this marker is within the range of your standard curve, within the range of quantification, straightforward. Just read the numbers. It will give you a concentration number. But next. But when you're at a low concentration that's outside of this range, you don't have a number. That's what's called detected but not quantifiable, basically, because it's outside of the range of this linear range for the standard curve. And as you can see, why the linear range has a limitation, you can see that these are all replicates. You can see the number starts spreading, right? That's because when the concentration gets low, your analytic precision is reducing. So that's why you cannot keep extrapolating to lower and lower concentration, because under this range, the statistic distribution changes. And this range, it's normal distribution, right? But this range is completely different story. It's a Poisson distribution now. But that's just geek talk. You can ignore that. so that's one problem. Now at a low concentration, there's no more linear relationship. You can't really quantify using standard curve. Next. And then there's non-detect. Because remember, we don't keep running the qPCR machine forever. We cut it off. Once you go through 40 or 45 cycle of growing, I'm going to cut it off. I'm going to finish my experiment. I want to read the data. So if the signal didn't come up to the threshold at cycle 40, it's done. It's a non-detect. But it's really, the thing is absent, not real. We don't know. So for non-detects, of course, there's no quantification. But you can't assume absence either. Okay, next. So, so what do we do with this? The previous solution, I already talked about this, right? You ignore the non-detect, but again, and the detect but not quantifiable. Again, this hardly seems like a good approach. Just intuitively, you're throwing away the data, or you arbitrarily assign a number to the non-detect and detected but not quantifiable. But what number do you assign to it? You're arbitrarily adding information to your data. You're not really respecting the data itself. So you can't say non-detect is 0. You can't assign detection limit to all the non-detect either, because that leads to overestimation, right? So there's a lot of uncertainty here. And then the third option is you can force the standard curve outside the RQ range, but like I just said, the data distribution and underlying these data, it's not valid for this approach. the standard curve should be extended. It doesn't work there. So then there are also statistics for sensor data. This is a gig talk, just in case there's some statistician in the audience who is wondering about this. It's not really applicable in most cases, environmental data, because environmental data, we get a lot of non-detect. So there's huge amount of censoring. A lot of stats developed for sensor statistics, they have a requirement. You have to have at least like 15% of a detection. Oh, no, 15% of non-sensor data means that 50% of the data needs to be within the quantifiable range. And a lot of sites sometimes don't have that. You have not a lot of detected but not quantifiable. So this thing is not really applicable in most cases. So what do we do for the human fecal score? What we did is we used underlying Poisson distribution to estimate all these question marks. Forward. So it's basically what we do is we use two different quantification mechanisms for the qPCR data, depending on whether they're quantifiable or non-quantifiable. If they're quantifiable, straightforward, just use the standard curve, right? It's within the range of quantification, use standard curve. If they are outside of quantification, if they're too low or non-detect, we use the Poisson distribution to estimate it. So the MPN, the Poisson distribution, MPN, you guys are probably more familiar with, it's like IDEX, right? Most probable number. So we use that, then we combine it to get a weighted average for human fecal score. And this is the equation for it. And this whole calculation is executed by Bayesian model. So Bayesian model is famous for integrating data uncertainty. So we can get an uncertainty measure and through the two population, sorry, the two distributions. Right. That's another game talk, but yeah, next. So I just talked about the human fecal score, right? What it really is. So another added benefit for the human fecal score is now it's mathematically defined. Everything is like you can just calculate it. Now you know how exactly you're going to use the data, and you can, knowing that, you can use this to optimize the sampling design so that you're collecting the right amount, right type of data. So like oftentimes when you go do a study, you're saying, okay, I want to know how this site compared to like site A compared to site B, but how many samples do I have to take? Can I go out and get 5 samples? If they're all non-detect, can I claim victory? I feel like the answer is no, but do I have anything to back that up? Not really, because the data, the backend, the data interpretation, I don't really know how I'm going to interpret it, how I'm going to optimize the design. So now knowing that you can use human fecal score to analyze the data, you can use human fecal score to evaluate what type of design is necessary for you to get the best out of your data to answer is sufficient. Let's say just using sample size as an example, how many samples you actually need to answer a particular question. So I will not go through the details of how you actually do this, but the end result is that this is an even deeper layer of optimization. This is like how many replicates, qPCR replicates you actually run for each sample. You can ignore that. I think Marisa likes that. She knows intimately about this. But this, let me just kind of focus on this side. This side is set sampling depth. That's like statisticians talk about sample size. It's presented as a percentage, but you can translate that to like 7 samples, 80 samples, or 100 samples, something like that. If your entire sampling, if your entire period you care about is 100 days, let's assume daily sampling, and that's what you have. So the dots here represent your study conclusion. Let's say your study conclusion is rank those sites. Remember the expert ranked 26 sites? Let's say I want them to get the same rank based on the human fecal score. Now I know exactly how I'm going to rank it. I'm going to use that to optimize my design. I want to be able to take fewer samples, but still get the same rank, the truth, like the rank based on human fecal score. So you don't actually need to take like the entire, say 100 or 200 samples, depending on whether you're looking at 100 days or 200 days. you can get all the way down to, if you're only running 2 replicates of qPCR, you have to do 93 days, right? But if you're running 3 replicates, and now you can actually get by 86 days. So on and so forth. Basically, and basically you can have a different study designs to achieve the same study outcome. So that gives the project manager or the stakeholders or the regulators have a flexibility, because sometimes it's more difficult for them to go sampling, but sometimes it's more difficult to run more qPCR replicates. But this allows you to, number one, get a sufficient design to answer a question. Number two, give you flexibility of managing your project. Which one you have more capability, you can use run more replicate or run more samples. And next slide. You can also optimize design based on the level of resolution or the level of certainty you want. So for example, I want to get the rank right 95% time. And this is the study design optimization need. So with the 3, if you're running 3 replicates, right, 86, let's say 86 samples, I want to get the answer right 95% of time. But if I'm willing to accept a higher uncertainty, if I get it like 90% right, I'm pretty happy. And because I don't have a lot of resources to do this project, but still I want to get a reasonable answer. 90% chance of getting the correct answer, I'm pretty happy about that. So in that case, you can reduce the sample size even further down to maybe less 80%, right? 80 samples, so on and so forth. So that's another layer of flexibility that would allow for study design optimization. Okay. So putting all these together, how we actually use it. So this is a hypothetical implementation study we published in the paper. So what you do is, we actually have 5 sites. For some reason, this graph only has 4 sites. So you start with the 4 sites. You use this, what I talked about, use the human fecal score to determine a optimized study design, right? How many samples you have to take and how many qPCR replicates you run. Then you generate all these qPCR data. Some of them are going to be in the quantifiable range, straightforward. Some of them is going to be non-detect or detected but not quantifiable. Then use the Bayesian model to calculate the site average concentration of this human marker. Now you have a single number for each site, and you just rank the site. B being the cleanest, and C being the worst, the dirtiest. And these ranges you see here is a certainty. Kind of, you can understand it as a standard deviation. In Bayesian stats, it's called credible interval, but it's the same thing, like standard deviation, just the kind of the precision you have. So this is an implementation of how you would use this to for site ranking. Okay. So what about real sites? What are the human fecal score in real sites in California? So these we calculate the human fecal score. Oh yeah, there's a change of name. It should be called score. From 7 sites in Southern California. Where they are is not important. So, but the study is ongoing, so I'm not going to disclose where they are. But the important thing is these are real size, and the human fecal score, as you can see, varied from about 10 all the way to 50 with the error bars, that credible interval I told you about. So just to put that in perspective, so we did a study for region 4 at a reference site, Leo Krill Beach. You guys probably very familiar with that site. It's a reference beach that I think, yeah, from a reference watershed. And so that site is actually really clean. So that site has a very low human fecal score. I think it's about 5. And then this is also the site that we did for region 4. This site is a site where we actually conducted epidemiology study. So we know the actual health risk. And that site had a score of about 37. So now you actually have a context of kind of where your sites are, again, kind of help enhance your ability to compare between sites. You not end up with a big table of qPCR results. How do I compare this site to another? You're reducing it down through the human fecal score so that you can present this sort of comparison in a more easy to understand way. Next. So just to summarize up, what I talked about the human fecal score, it's a simple number, so it's fairly straightforward to allow comparison. The second thing is it respects the data. It uses everything. It doesn't ignore, non-detect, or those samples with question mark where you can't really quantify them. It doesn't add anything. It adds nothing. So it doesn't force a detection limit or arbitrarily assign a 0 to non-detect. So it's not adding information to the data. And also respects the underlying data distribution. If the data follows a Poisson distribution, I'm going to use Poisson to estimate the concentration. If the data follows a linear range, standard curve, I'm going to use standard curve to estimate the concentration. And also it integrates the uncertainty in the data through Bayesian modeling. So, and then because it's mathematically defined, it's a lot easier for experts to actually implement it because it's not descriptive or subjective anymore. They're just calculating following a mathematical formula. So it's built on formula instead of narratives. Then finally, it's not a standalone approach. It's actually integrated through a standardized US EPA qPCR method that it's kind of in the pipeline of being published in the Federal Registry. So that's the standard method we're using. It also allows you to a formal way to optimize the cell design. So just a few words on what does this mean? What can this help you in terms of the qualified management? And the first point is reiterating now it's a standard method for site assessment to see how polluted your site are, specifically with the human fecal contamination, not how polluted is it in terms of FIV. Okay, that's a big distinction I want to make. And also the human fecal score is general enough that this framework can be applied to the cow marker, also the gull marker, and so on and so forth. So you can call it the cow fecal score or gull fecal score, whole franchise of scores. And then it's also applicable to other technologies. I've been doing a lot of digital PCR talks. I kind of like, it's one of my favorite little child. So it's also applicable to digital PCR as well. And in terms of potential application, this goes back to the first slides where what are the questions you have, right? A lot of time your question is related to comparing sites. So comparing the site before and after BMP effectiveness, comparing the sites within your jurisdiction in terms of where you're going to put effort to solve a problem first, or in terms of outside of California, the CSO combined sewer overflow, it's a big deal. There's consent degrees of fixing that. So how do you show you're actually in compliance? That's another application at the national level. And then finally, when you have when you can demonstrate that your human source is really under control, you really don't have much human contamination in the revised recreation criteria. It says that perhaps you should consider alternative management strategies through QMRA to establish a site-specific criteria. So that concludes my presentation, and thank you so much for your attention. And this is my contact. If you want a copy of that paper, just contact me. I'll send you a copy.